The overall goal of this procedure is to assess the antifungal activity of the primary human immune cells in real time using fluorescent Aspergillus reporter conidia and live cell video microscopy.
This method can help to answer key questions regarding the immune responses against fungal infections in specific patient populations, in particularly the innate immune response occurring early upon exposure to fungi. The main advantage of this technique is that it can be used to identify specific defects in innate and antifungal immunity, such as phagocytosis, cell migration, killing, and inhibition of fungal growth.
To begin, streak Aspergillus fumigatus strain AF293 carrying the fluorescent DS red reporter gene over agar prepared in 2 T75 flasks. Incubate the cultures at 37 degrees Celsius, 5% CO2 for 7 days to obtain approximately 10 to the 9th conidia. After the incubation, immerse the cultures in 20 milliliters of PBS with 0.05% tween 80 and collect A. fumigatus conidia. Then, using a 40 micrometer cell strainer, filter the resulting suspensions into a 50 milliliter tube to remove hyphal fragments. Once the suspensions are filtered, centrifuge them at 805 times G for 10 minutes. Then discard supernatants and resuspend the pellets in 20 milliliters of sterile PBS. Finally, pool the samples and centrifuge the resulting suspension.